Hey, good morning, everyone, and welcome to the 2020 edition of the Royal Tyrrell Museum Speaker Series. Today, the Royal Tyrrell Museum and its cooperating society are proud to present Dr. Sarah Otto. Sarah is a professor in the Department of Zoology at the University of British Columbia in Vancouver. Sarah is originally from New York City. She obtained her bachelor's and PhD degrees from Stanford University in California. For her PhD, Sarah studied evolutionary dynamics by investigating the role of DNA recombination, the number of chromosomes, and non-random mating in sexual organisms. After completing her PhD, Sarah held postdoctoral fellowships at the University of California, Berkeley, and the University of Edinburgh in Scotland prior to being hired by the University of British Columbia in 1995. Sarah's research focuses on determining how biological systems evolve using models to investigate the selective forces acting at the genetic level and on reproductive strategies. Complementing this approach, Sarah is experimenting with yeast, watching how they evolve to test evolutionary theories. Sarah helped found the Canadian Society of Ecology and Evolution and has served as president of the Society for the Study of Evolution, as well as on numerous other councils and boards. Today, Sarah will discuss how the evolutionary trajectory of life is being altered in an increasingly human-impacted world, altering which species survive and which go extinct. So without further delay, I present you Dr. Sarah Otto.
Well, it's really a pleasure to be here. The Tyrrell Museum is on the bucket list for evolutionary biologists to come visit. And of course, I've already been here with my young son, but it's such a pleasure to be back. So thank you for the invitation. I forgot to ask Francois what the proper acknowledgement, land acknowledgement is in Drumheller. But I would like to acknowledge that we are gathering today on the traditional territories of the Blackfoot Confederacy. This is from Calgary. Is this similar here? Anyway, in the spirit of reconciliation and moving forward together, I would like to acknowledge the original traditional territories of the First Nations that lived in this region of southern Alberta. So today, I'm an evolutionary biologist. That's what Francois mentioned. And I am interested in understanding how evolution led from a single cell organism 4.1 billion years ago to the enormous diversity of life that we see on this planet. As he mentioned, I'm particularly interested in understanding the kind of twists and turns of evolution, why sometimes evolution leads to a remarkable diversity of life and sometimes it's really twiggy. Why organisms sometimes are sexual and sometimes are asexual, have large genomes, small genomes. What's interesting about this evolutionary process is it hasn't led to a single solution. There is no optimal way to make a living on this planet. This diversity tells us that under different circumstances, evolution will take different paths. And it's that process of understanding why sometimes do we get small organisms and sometimes large organisms? Why does sometimes the genome gets compact and smaller and smaller and smaller, and other times huge? There are organisms, ferns, with 1,000 copies of their genome in their cells. Why? Under what circumstances? And so I use math to try and tease apart to understand those alternative trajectories. And as Francois mentioned, both yeast experiments, but also looking at the shape of the tree of life, because the shape of the tree of life gives us clues about what has led to evolutionary success, to speciation in the past, and what has been evolutionary dead ends, because we see branches missing, places missing, and that changes the shape of the tree. So that's just a little snippet of what I do. I think I'm going to point out that while we think of the evolutionary tree of life as being 4 billion years old, it is actually much more evolutionary time that, of innovation. What do I mean by that? If we summed all of the branches of the evolutionary tree of life, that would represent how much kind of evolutionary experimentation has been done in the different species that have lived on this planet and that are alive today. And we don't know that exact number. We don't even know exactly how many species are living on this planet. Guesses are around 10 million. But if we sum the shape of the tree of life over those species that are alive today, I get that there's been 10 to the 15 years of evolutionary innovation independently on these different branches of the evolutionary tree of life. 10 to the 15. I'm a mathematician, and yet I don't even know what 10 to the 15 means. It's just a very, very large number. So I Googled, what does 10 to 15 mean? For comparison, in the entire published book of human history, there are only 10 to the 14 letters. So this is more time of evolutionary progress across the tree of life than letters in all published literature, just for a sense of scale. And I raise that because evolution, what is evolution? Evolution is a process of mutations appearing, leading to genetic variation within species. Some of those variants allow individuals to survive and reproduce, whereas others lead to death and sterility. That process of mutations arising, being tested in the environment, explains why we see amazing adaptations to the world around us, why species have evolved to tolerate heat and acidity at deep thermal vents. The high Arctic, why I even just read about a species of bacteria that is growing near Chernobyl, where we had the horrific nuclear accident in 1986, and they've evolved to take the energy from gamma radiation and turn that into life, to continue to propagate and reproduce. So pretty much all environments on this planet are inhabited, and it's simply this process of mutations appearing, and those that better allow an organism to better match its environment are the ones that survive. And so as an evolutionary biologist, when I see this tree of life, I see a treasure trove of 10 to the 15 years of innovation that the biological world has undergone. And so in that context, I want to talk about how we as humans are changing this tree of life in a permanent way. How are we shaping the evolutionary tree of life? So that's going to be the topic of my talk today. And some of the research that I and my students do is peppered in here a little bit, but I want to say that by and large, this was an overview of the research that many, many, many different evolutionary biologists have done. And I wrote an overview paper, Adaptation, Speciation, and Extinction in the Anthropocene. If you write to Francois or to me, we can send you a copy of this paper, which has the actual references and everything to the studies that I'm going to talk about. So let's just start by saying, when I was a kid, I don't think we had a good sense of how we were impacting the globe. We knew how we were impacting our yard, maybe our city, but we didn't have a sense that we were impacting the globe. The planet felt too large, too infinite. And so as a child, I remember we used to throw things out of our garbage out of the car window as we were driving by. And that stuck in my head because whatever happens to that? But the view then that my father responded when I asked, why are we doing this, was that it'll go away. It'll get recycled. It'll compost. The earth will take care of it. That the earth was capable of basically tolerating all of our insults to it. And we don't think that anymore. The population size has now grown to over 7 billion. And our footprint, which is the way that we measure how many resources we extract from the environment, that has been growing too. So the sum total of a burgeoning population size and burgeoning demands on the planet has led to massive changes across the globe. Just as some statistics to ground us, 75% of ice-free land on the planet has been impacted, and that can be that is looking at pixel by pixel across the globe, whether there's roads or agriculture or cities or what have signs of human impacts on the planet. So 75% of the land has now been impacted. 67% of the global biomass of predatory fish has declined. 25% of the surface of the earth is considered degraded by human-caused erosion, pollution, compaction, and salinization, salt addition. Temperatures have increased over the last century by 0.85 degrees Celsius. Sea levels have risen by 0.2 meters. Oceans have become 26% more acidic. The point, I'm sure you've seen some of these numbers or heard these tolls on the planet before, but the point is we're having a global impact. There is no species living on this planet whose survival versus extinction isn't impacted by us. And I don't think my premise in this talk, my hypothesis in this talk that I want to discuss with you is that there has never been another species in the entire planet that has changed the course of evolutionary history in a single century as much as humans have. So just as more background, I want to talk about how most of the time when we talk about human impacts on the biological world, we use how are we affecting the number of individuals in wild populations. So for example, this is the Living Planet Index that basically takes all scientific studies that have looked at the same population over time to see how are humans impacting the population size of wildlife. This comes from a studies of over 3,000 vertebrate species, multiple populations. And since I was born, the population size on average of these vertebrates has declined by more than 50%. So these global impacts that we're having is leaving less and less natural environment, and that is having measurable impacts in reducing the population sizes of many of the species on this planet. There was a similar study that got into the news recently that recorded just how many birds are there now relative to how many they were in 1970. And the declines were missing 2.9 billion birds. And I don't know, I remember just barn swallows being everywhere. Bats are similar, major declines. I remember bats being everywhere. I don't see barn swallows very much. I don't see bats very much anymore. How many people here are birders? And citizen scientists in general? That's great. Well, I'd like to say thank you because this was possible. Our understanding of these changes in biodiversity are possible because of citizen scientists. These numbers come from tallying up trends in two ways. They come up from two ways, either tallying up trends from annual breeding surveys, Christmas bird counts, etc. And those have been going on for so long now that we can understand how bird numbers have declined, but it also is really technologically, we're getting better at understanding biodiversity. This is a radar map. We don't have one in Canada, but there's one in the United States. And all of these radar points, you can go to this website, birdcast.info, live migration maps, and see how many birds are in the air, because it changes the radar in a way. They know how fast birds move. They understand this is not a cloud. This is a group of birds migrating. And those radar counts give a similar picture of decline of birds. We've only had that for 10 years, that type of data, but the two match perfectly. So populations are declining globally. And what I want to turn to now is discussing how are these global impacts on biodiversity affecting the processes of evolution. Selection along lineages that changes organisms over time. Extinction, the lopping off an end of a branch, or the speciation process by which one lineage becomes two. So that's going to be the three sections of my talk. And I'm going to start with human-altered selection pressures. I grabbed this image from the web of a escalier, a trellis. Here, this image in my mind reflects how we're altering selection. These days, it's not who survives and who reproduces isn't only about who is better able to obtain resources in the natural world and who is best able to avoid predators, but today, more than ever before, who are favored, what is selected, are the organisms that best tolerate the land development, harvesting, species introduction, and environmental changes caused by us. Darwin wrote and contrasted natural selection and domestication as two forms of natural selection and artificial selection leading to domesticated species as two distinct forms of selection. I would say that that is increasingly blurred as humans are shaping selection across the globe. So I wanted to, I'll spend a little bit of time diving into that because I don't think we think about it enough. How are we changing the selection pressures and evolution of species around us? So I'm going to talk about the many, many different ways in which we've done that and the types of evidence that show this escalier process for selection. First, selection to survive in our built environments, around cities, around roads, around power lines. Animals and plants have to survive. One of the major sources of mortality for birds and bats is collisions with buildings, automobiles, and power lines. 800 million birds in the United States and 60 million birds in Canada annually. Those are the deaths, and so we can ask, what about the survivors? Who's surviving? And studies are increasingly showing that those survivors behave differently and they are morphologically different. For example, great bustards there's migratory great bustards, and there's ones that stay put, that stay in their local environment. And the number of great bustards that stay put has increased from 17 to 45% in 15 years, in large part because of the high risks of migration that these bustards face, including collisions. Another study looked at cliff swallows and showed that the swallows, this is crazy, but the swallows near roads had wingspans that were declining over time, 3 millimeters, I believe, in 30 years. And they also were able to document that the roadkill, that the cliff swallows that were killed on the road were the ones with the longer wings. I don't know if anybody knows much about aerodynamics, but it's very hard to maneuver with long wings. Shorter wings, it's easier to outmaneuver a car. And this was evidence that indeed our cars were selecting for shorter wing cliff swallows. What are the implications? We don't exactly know. More and more species are changing to survive in our environment, but who knows how that's affecting the ability of cliff swallows to migrate where they need to migrate to their breeding grounds. How is it affecting their ability to catch insects? They're evolving to us, not to their natural world. So I've talked about mortality, but sometimes our built-in landscapes are actually opening up new niches, new opportunities for selection to operate. One crazy example is that Culex pipiens, a mosquito, has evolved to tolerate or to live underground in our subways and other tunnels. And what's crazy about this is normally there wouldn't be any mammals or animals to bite down there, but because we're down there in their subways, they have a great source of blood. And these mosquitoes have not only evolved to live underground, they've also evolved to like human blood more than the bird blood that they used to taste. So that's another example of how selection to survive in our built landscapes is altering. You can think of it metaphorically as altering the fitness landscape of the world. That fitness landscape is now a built one. A second form of selection where we're causing massive changes in the biological world has to do with what we hunt and what we eat. And this is a very classic, some of the first papers on this had to do with how our hunting was selecting for the survivors to have the traits that kept them out of our traps, that kept those animals out of our crosshairs. So for example, silvercoat foxes were extraordinarily popular at the beginning of the 1900s, and they were extirpated from the eastern coast because of their popularity. We were selecting against silvercoat variants in foxes. The ones that survived did not have that. Similarly, there's a strong selection because of poaching against longer tusks in elephants. And one study that documented the effect of poaching on elephants found a decrease in tusk length of 25% over a generation. That's how much mortality was going on. So there's a second thing about human-imposed selection pressures. They tend to be really strong, especially in cases like this. 25% change over a generation on an evolutionary timescale, that's lickety-split. Normally we think of evolutionary change over thousands or tens of thousands of years. This is one generation. That's enormous speed of evolution. Another, more examples of are the impact of hunting and harvesting. Of course, fishing is probably the best documented examples of this, where fishing selects for less catchable fish. And that has led to a decline in body size in many species that are are captured in gill nets and other large netting. So there's many studies of smaller body size evolving in fish. But there's even crazier ones where there was a study that showed that fish that were subject to hook and line fisheries had evolved smaller mouths, so that the hooks would be less likely to capture those fish. The survivors had smaller mouths. The fish that had larger mouths were taken. And it's not only changing morphology in those ways, but it's also changing behavior. Rainbow trout, in a recreational fishing area, had evolved to be faster and bolder, basically to be able to either, I don't know how much of that is to be able to avoid the hook or be able to get off of the hook once hooked, probably more that. But anyway, faster and bolder rainbow trout evolved in response to our fishing. A third area where actually I think we know less about this, but it's probably going to have a bigger impact globally, is that we're changing which species are in contact with each other. We're changing the biological communities through introduced species, invasive species, but also with climate change. We're shifting which species are in contact with one another. Just as one example of this kind of impact, introduced honeysuckle. Cardinals love introduced honeysuckle. So the cardinals that are in the best condition, the brightest red, they go and they take these territories and they nest in them. But it turns out that predators can better get them in honeysuckle. So these very bright red cardinals are now nesting in a primo spot for predation, and they're being killed. This is reversing the direction of selection, whereas before the highest quality males could hold the best territories and red color was strongly selected, and now it's reversed. And we're seeing the loss of red coloration or declines in the red coloration due to this increased predation, because they had not evolved to avoid honeysuckle, to avoid this predator zone. Of course, if we look around our cities are not natural environments. And they are patchy environments where species can't grow everywhere. And that creates more selection pressures for an interesting phenomenon. Plants in many of these urban environments aren't setting their seeds out as they used to. They set their seeds right where they're growing. Because of course a plant like this is Crepus. If it sets its seeds, so they have little pappuses, little umbrellas that float in the air, in the wind. And those large pappuses that would take their seeds or further distance are now landing on concrete. So the ones really small pappuses that don't have those umbrellas are landing right underneath the plants, and that allows them to survive in these highly fragmented environments. Because of climate change, migration, so that's avoidance of roads, and concrete is selected for less dispersive seeds in our cities. Migration timing has shifted due to climate change, and the scale of this is pretty large, 2.3 days per decade. And in pink salmon, another study showed 2 weeks earlier migration due to climate change in 40 years. And in this particular case, they even had a gene that they knew was involved and had risen in frequency that was associated with earlier migration. And the last example that I want to give of how we're changing this fitness landscape in which organisms are evolving is we're even shifting the way they reproduce. When we domesticate animals and plants, our domesticated animals and plants recombine more. That increased recombination is favored after a period of strong selection because more variants are produced during that process, and that leads to the increase of genes that favor recombination. So these species are recombining more. Our microbes, many of them, are mutating more because of selection due to antibiotics and other human-imposed selection pressures. And even the genome size is evolving in response to humans. Many of our crops, from sugar to wheat, strawberries, are polyploid. Humans have selected for larger genomes, oftentimes because of an association with a larger fruit size. So we're changing the very way that organisms live and reproduce, shifting their inheritance. But of course, ones that are responding, the ones that are staying along this trellis, are keeping up with our environmental changes, but not all species will be able to respond fast enough to keep up with the human-caused selective pressures. So one thing I just want to do a little bit of a brief aside of how we're studying this, both in my with math and with experiments in my lab. And the question that I want to know is, if a population size is declining because of human-altered environments, will that lead to extinction, or will the population be able to keep up, and will mutations spread and allow the population to survive? One thing that we showed mathematically was it's actually evolution is hampered. It's like it's handicapped by declining population sizes. When a population is declining, you have a poor sample of the parental generation. There's a lot more random noise because you have a poor sample of those parents. And we showed analytically that this increased amount of randomness due to poor samples leads to a smaller chance that good mutations, adaptive mutations, can fix, and a higher chance of deleterious mutations can fix. So not only in a declining population are there fewer mutations, but the good ones have less of a chance of making it because their kids may just not happen to carry them. If they do carry them and the population is able to recover, we call that evolutionary rescue. So that's one of the key areas in evolutionary biology right now, is to understand when this evolutionary rescue happens versus when extinction happens. I'm going to just briefly talk about some experiments that my students Eliza Gerstein and Jasmine Ono did, looking at rescue mutations in yeast. This is the yeast that makes our bread and wine, but it is also a great organism to experiment with. They replicate like every 90 minutes, which means that we can watch evolution over the course of a week. So these are short-term, days experiments where we put them in environments that are just a little bit too tough, a little bit beyond what they can tolerate. We've done this with a fungicide, nystatin, and with a heavy metal, copper. And then we basically do this so many times. We put them in little wells where we put this copper or the nystatin, and we put about 100,000 cells, and we see what happens. The cool thing, compared to when I was a student, is when we do get cells surviving, we can then sequence the entire genome of yeast. It's a teeny tiny genome. It costs about $100 to sequence the whole genome. So we can find out exactly what evolved and allowed them to survive in this changed environment. And most of what we were doing this for was to understand the genetics behind this. And I'm not going to go into that, but I do want to tell you how often did they get rescued or go extinct. In this particular experiment, what do I mean by rescue? You see these little white clouds? That's actually settled yeast. Though in those wells, these are identical to begin with, but in those wells, a mutation happened that allowed the yeast to survive. Those got rescued, whereas these ones, nothing grew. They went extinct. So overall, out of our, 288 lines, 35 mutant lines were rescued in nystatin, and 34 mutant lines rescued in copper. One of the aspects of this that we were interested in was whether or not the nature of the environment makes it easier or harder to be rescued. So we could look at, would those mutations have rescued these lines if we accounted for not just human-caused environmental change on one axis, but the many ways in which we change the environment. This experiment gives us some insight into that. If we just change nystatin, we know that these mutations are beneficial. And in this cartoon, I'm picturing that as this is the optimum, the higher growth, and this is lower growth, and they start down here, and our mutations are improving their ability to grow in nystatin. When we measure them in copper, those mutations, by chance, the genes that were affected, improve how they grow in nystatin and in copper. So if the environment changed in this particular way, there'd be lots of rescue mutations. But there was another combination, salt, where we saw none of the mutations that were beneficial in nystatin were able to tolerate salt very well. So depending on the complexity of the environment, rescue may be very, very hard or very easy, and we still don't have a very good sense. I can't predict very well when it will be easy and when it will be hard. And that depends on whether the genetic changes tend to align for those environmental changes, like in copper and nystatin, or aren't aligned, like in nystatin and salt. The other thing that Jasmine did was those experiments were done with haploid yeast. Haploids means they have one copy of every gene in the genome. But we also have diploid yeast. They have two copies of every gene in their genome like we do. The diploids never rescued. She did this over and over and over again. She did it in little wells in big flasks, and the diploids never were rescued. The haploids were, but not the diploids. And that's because what the diploids needed were always masked. The beneficial mutations were always masked by the other copy, and the diploids were never rescued. And this indicates that there are going to be species and types of species, like haploid species, that might be in certain environments more likely to survive our changes, and others that are less likely to survive. And so that I want to turn next to thinking about that, about how we're lopping off these limbs of the tree of life and shaping the tree of life through extinction processes. I'm sure here in the room you're familiar with this extinction crisis, as it's often called, or biodiversity crisis. There was an IPBES report. IPBES is an international panel that analyzed all of the data on extinction rates and came to a global assessment approved unanimously by 124 member countries, and that was just last year. And one of their main findings was that if you calculate how many species are threatened with extinction globally, and remember we don't even know all of the species that are on the planet, so they had to say this fraction of insects is threatened with extinction that we know of, and this is our guesses about how many insects exist. So they had to do those type of calculations to come up with this number. But their conclusion is that nature is declining globally at rates unprecedented in human history. And the rate of species extinctions is accelerating with grave impacts on people, on the world now likely. And they ask us to argue for and make the transformative changes needed to restore and protect nature. So I just wanted to give a shout out to where those numbers come from. This million species comes from experts that around the globe are studying different groups, from bony fishes to cycad plants, and asking how are those populations doing? Are they declining? Are we losing populations? How at threat of extinction are they? The color coding here, red is critically endangered, orange is endangered, vulnerable. The greens are healthy and of least concern. And these overall tabulations now of thousands of species have allowed us to conclude that over 1 in 5 species of vertebrates, 1 in 5 invertebrates, and 1 in 5 plants are now at risk of extinction due to human-caused changes. Extinction rates compared to the fossil record or compared to phylogenetic trees of life are about 1,000-fold higher than they were. Here in Canada, this is a map of where our endangered species live. This is high overlap of endangered species, low overlap, and it's not a surprise. They live where we live. Our endangered species are found where we've impacted the country the most. And they include things like the little brown bats, due to we think of it as white nose syndrome, but think of it too as an invasive species that we introduced from Europe. It is a human-caused extinction risk. Bank swallows, cliff swallows, and many other insectivorous birds have been declining, and we don't exactly know why, but there are myriad threats that they face, including declining insect populations, as well as climate change and mortality due to buildings and feral cats. This is Eulachun in the Fraser River, a really important fish to the First Nations in the BC area. Candlefish, it's also called, declining because we like to straighten rivers, we like to dredge rivers, and that eliminates their habitat. The southern resident killer whale, again, multiple pressures from increased boating traffic, but also decreased Chinook salmon. And just to point out that if we tabulate what are the pressures that these species are facing, we find a similar picture either globally, this was done by WWF, or within Canada, this was done by Jenny McCune, a new hire at Lethbridge, so she would be a great public speaker. And basically, it's the same old things that we would expect. Human disturbance, invasive species, resource consumption, pollution, transport. All of these ways in which we're changing the environment are listed in the Canadian Species at Risk Act as a threat to our Canadian species. I'll point out climate change is down here. It's actually not, it's one of the lower threats facing species today. But I think that that's just a matter of time. We're seeing increasing temperatures, and that's leading to many species facing more and more climate change associated mortality, like the white pine beetle infestation that was associated with climate change here in BC and Alberta. And one of my colleagues at UBC has looked at how fast is climate changing for forestry in BC, and documented that the best environment for a tree to grow is moving north 1 kilometer a year. Trees can't get up and walk 1 kilometer. So we're seeing an increasing mismatch between where trees can grow and thrive and where their habitat is for them. And that's led her and her team to help advise BC foresters so that they change their reseeding practice. It used to be you could only replant seed from a local area, and now they're allowing more seed to be taken from further south to increase this chance that we're just give them a head start in this adaptation to a changing world. So we don't know yet, but it's been estimated that extinction risk will accelerate with future global temperatures. And Mark Urban estimated that 1 in 6 species, given changes in ranges of these species, would be under threat of extinction if we continue the business as usual climate projections along that trajectory. So 1 in 6 species, and that's in addition to the species that I've already mentioned that were at risk from other factors. So which species are we lopping off this tree of life? We know that there are the species like that silver variant fox that we admire and we hunt, and that has led over the last century to many of the extinctions of species, including sea mink, passenger pigeon, and the great auk, because we over-harvested them. Over longer periods of time, over the past 50,000 years, we have strong evidence that human-induced extinctions are the larger-bodied animals. We've lost half of the larger terrestrial species, and I think many of them are displayed in your museum. Studies have shown that species that have a smaller range are more prone to extinction now. Woody plants are more prone to extinction. Tropical plants more prone to extinction. Dioecious plants, those with separate males and females, also more prone to extinction. So studies are accumulating saying who is more at risk of extinction and how are we changing the evolutionary tree of life as in response. And then in a nutshell, the answer is that the widespread, generalist, dispersive, easily mobile species and human commensals, the ones that can live and tolerate our built environment, are on average less prone to extinction in this human anthropogenic era. And the last topic I want to turn to was speciation, because that's another key part of evolution. And many people, I've heard people say, well, yes, humans are causing a higher extinction rate, but won't we also be increasing speciation rate and eventually everything will be okay? And I want to talk about this. What do we know about how humans are impacting the rate of speciation? Actually, we know very little is the bottom line answer, but we know some things. We know of cases in which humans have created new niches. That mosquito I already mentioned in the subway system is one. But the famous case of Ragelius pumminella that switched from hawthorns to apples is an example of where we created a new niche, apple farms, and that allowed this particular fly, the variants that went to apple that detected and chose to live on apple trees, those became isolated for those individuals that preferred hawthorn. Cereals, another example, the increase in wheats and rice and other cereals has led to speciation in the fungus underground that attack those cereals. Mine tailings are another great, horrible example. When we pollute the earth, many times we see new species form on those mine tailings because they can tolerate because mutations have allowed them to tolerate the high copper or other heavy metals, much like in our yeast experiments. But then they're also selected to not mate with the plants around them because that would then eliminate or dilute the adaptations to those mine tailings. So sometimes we make new niches and that increases speciation rates by producing new environments for species to colonize. Sometimes we do the exact opposite, where we take a fairly finely differentiated environment and we collapse that. The classic example of this is in Lake Victoria. Eutrophication and agricultural runoff has led a clear lake to become a muddy lake, opaque lake. And cichlid fishes, there are hundreds of them in this lake. And they mainly were differentiated by sexual selection. Who mated with whom? Their colors, incredible coloration differences. And we've lost many species in this lake, basically because they can't tell each other apart anymore. Another example in BC that my colleagues worked on was in Enos Lake, where we introduced an invasive crawfish, and that crawfish basically completely changed the shoreline of the lake. And whereas we had 2 species in those lake of stickleback, when that shoreline was devastated by the crawfish, they started hybridizing. Their nests were in different places, and now they are all mating with one another, and we just have one hybrid swarm of sticklebacks, not 2 anymore. So that's collapse niches cause the speciation rate to go down. The other thing we're doing is we're fragmenting the environment, making populations more likely to be isolated, and that can increase speciation rate. Panama Canal, for example, has isolated tamarins on either side, and those are now reproductively isolated to some extent. But of course, we're doing the opposite too, and that is rather than fragmenting, we're moving species and taking what was geographically isolated and bringing populations together. And that can have a complexity of effects. For example, when we've moved Spartina alterniflora into California, that led to a hybrid swarm and a collapse of 2 previously isolated species that were on different continents and marching along on their evolutionarily independent ways until humans got involved. But in other cases, those hybrids then form new species, and that happened in Europe with Spartina anglica, a very invasive form of Spartina. The bottom line is we know almost nothing about speciation and whether or not humans are causing increases or decreases in the speciation rate overall. We can study this in the lab too, and many people wouldn't think you could study speciation in the lab, but as I said, yeast evolved so quickly that we can take those lines that adapted in our fragmented wells and then cross them and see if we brought them back into contact, how would their offspring do? And basically this is a cartoon picture of what we're studying in the lab, where mutations accumulate in our little yeast independently over time, but when we cross them, there's new genetic combinations that were never evolutionarily tested. Big A has never interacted with Big B. That mutation and this mutation have never seen each other, and there's no guarantee that they work well. And indeed, speciation is about this process of the hybrids doing poorly. That from horses and donkeys to any crosses are good species. The hybrids either don't get produced or are sick or dead when they are produced. So we wanted to know, does this happen? So Jasmine did these crosses and measured the fitness. And what I'm going to show you is these cartoon pictures or these images of her findings. So she took her different mutations and measured their growth rate. So this axis is fitness, and this is just, we arrayed them in this way. The ancestor is down here. The single mutants, as I mentioned, are these corners. And then she crossed them and got the double mutant. And so her question was what happens overall, and she did this with many different mutations. And what she found was that the double mutant was often depressed in fitness. The hybrid was less fit than the 2 single mutants. She found that in 2 of 6 cases where the hybrid was less fit than either parent, and in another 2 cases where it was less fit than one parent. So even though this experiment was only over the course of days, the hybrids formed are less fit, and we're starting to see this process of speciation in the lab. Species aren't everywhere. We have millions of species on this planet, but we don't have trillions of species on this planet. And if it's that easy to get speciation over the course of a week in yeast, we'd be overrun with different species of yeast. So this makes me wonder if there's things about this experiment and things about how humans are impacting evolution that may affect the speciation process. And so I'm going to show you a cartoon of what I mean. I think that we saw speciation because we were selecting so strongly that we had huge effect mutations that allowed them to survive in these new environments. So if this was the original environment, we were selecting for these big honkin' mutations. And when we cross 2 mutations that both work well and bring the yeast closer to their optimum in this environment, when we cross them, we'd get overshooting. And so that the hybrids are less fit than either of the parents were. And I contrast that. If we had selected very slowly, then we would have accumulated a whole bunch of different mutations. And if you hybridize these 2 parent populations, you'll get a mixture of some of these arrows, some of those arrows, some of these mutations, some of those mutations, and it would still be very near that optimum. So I think that strong selection, and especially these rescue mutations, these very large mutations, may very well be a speciation engine. And I don't think the ticking of the speciation clock is constant over time, but much more likely during periods of rapid environmental change. And I've been working with a student, Ryo Yamaguchi, to show that, that this cartoon picture holds up when we actually do models of mutation and selection and speciation. Humans, I do think, can, under some circumstances, be an engine for speciation. But there's a really key point I want to make here. And that is, when we lop off, due to extinction, branches of the tree of life, we're lopping off a lot of evolutionary time. We're lopping off millions of years. And then if we are increasing speciation, we're increasing the rate of production of baby species with almost no evolutionary time separating them. These are newly formed species like the yeast in our lab. They're genetically identical, except at 2 spots. They are not a treasure trove of evolutionary innovation. So we're losing the trunks, and we're getting, if speciation rate is going up, we're getting a lot of this, these sports of very short, very little differentiated species at the tip of the tree of life. And so I'd like to close. We don't know all of the answers. We barely have scratched the surface of understanding how humans have impacted evolution. But I do think that this is worth thinking about. Humans have altered the course of evolution, and that is leading to a legacy of an altered biological planet. It's that trellising, it's the species that can survive and that are responding to selection along the lines that we impose rather than what nature's imposing, that are going to be the successful species in this planet. Human activities are leading to the loss of large-bodied specialist narrow-range species, as well as species that are otherwise vulnerable to humans. And as I mentioned, even among the surviving species, we are the common selective pressure to tolerate human activities rather than the natural selection pressures they faced before. And by moving individuals around the world and homogenizing environments, we may also be reducing the rate of speciation and extending the time for and if we are doing that, we may be extending the time for the planet to recover after this extinction crisis, just if there's fewer species. And even if we are increasing speciation, we're leaving a legacy of very similar new species. So I want to reflect in ending back to the IPBES call for us to think about transformative changes needed to restore and protect nature. The hopefulness in me comes from humans are incredibly resourceful, And we can make change, and we can make change remarkably quickly. So I think working together to reduce our footprint on the planet, to work with land trusts. Is there an Alberta Land Trust organization? Yeah, and I work with the Nature Trust of BC as well sometimes as with NCC. Because what we can do with these land trusts is, remember that picture of Canada? Well, we can buy land and protect it for nature in those regions where there's the most species at risk, and where we can also purchase land that allows connectivity and kind of reverses those fragmentation pressures on the species around us. Anyway, I would like to end with a hopeful picture, because I think together, and I know that the youth of today are really want to see a natural world for themselves and their children. So that is what leaves me hopeful. Thank you.